Today is the 26th October 2021, and welcome everyone to the WebEVTalk. At the moment, we are on the USA and Asia Pacific time zone. So it's on the 25th October in the USA. So it's my pleasure to welcome Hakho Lee. He is Associate Professor in Radiology, Massachusetts General Hospital or Harvard Medical School in the USA. He currently directs the Biomedical Engineering Program at the Center for Systems Biology and is a Hostetter MGH Research Scholar. Dr. Lee has extensive experience in EV analysis. His research focuses on developing sensitive, cost-effective, and user-friendly tools for EV characterization. So, for instance, by integrating principles of nanomaterials, biophysics, and engineering, his group advanced new analytical technologies, including the nPLEX, iMEX, iMER, and CEA. So I'm really excited to listen and learn about this new technology. And some of these platforms are now being translated for clinical uses. So the title of the talk is Development of New Technology Platforms for Extracellular Vesicle Analysis. So I'd like to thank Horiba Scientific, who has been supporting us since April, running the WebEVTalk. And this session is also supported by the Horiba Scientific.
Okay, thank you so much for the invitation. And this is a great platform, actually. So Carolina told me that she's been doing this for about 100 talks so far. My goodness! I mean, if you haven't heard all those talks, then you will be already EV experts by now. So thanks, Carolina, for this great effort. So I'm here at Mass General Hospital, and then I'm leading the biomedical engineering program and developing these new sensors for EV analysis. So we started this research about 10 years ago or 12 years ago almost. And we've been developing these new sensor systems for in vitro EV diagnosis. So this is one really brief intro slide. So most of you are now familiar with the liquid biopsy or circulating biomarkers. So they really offer the new opportunities, in cancer management. That includes the diagnosis, prognosis, and treatment monitoring. And compared to the core tissue biopsy, this getting the circulating biomarkers, they're much easier and non-invasive, right? So you can do the repeated biopsy with the minimal complications, so that actually can give us the window to look at the temporal evolution of the tumor. So what is happening before the surgery, for example, and what is happening in tumor after the surgery, and then during the treatment. And so this shows the 4 examples of these circulating biomarkers. Of course, there are the shredded tumor cells or circulating tumor cells, extracellular vesicles, cell-free DNA or the circulating tumor DNA, and microRNAs. And the main reason that we've been focusing on one of the main reasons, I should say, focusing on the EVs or extracellular vesicles, is that they're abundant compared to the other detection targets. So the concentration of EV is about 10 to 10 particles per milliliter of blood. So it's much easier to detect compared to the very rare targets. Of course, there is a caveat which I will explain later in this presentation. So they're abundant. I mean, the EVs are abundant, but there are challenges, of course. So this picture actually says the challenge, in one picture. So here you're looking at, this is GBM, the cancer cell in culture, and you're looking at the single cell, one cell, and the diameter of this one cell is about 20 micrometers. So they're relatively big, 20 micrometers in diameter. Extracellular vesicles that we want to measure is way, way smaller than the cells. So basically, these tiny particles that are going on and off on the cell surface. So when you zoom up the size of these particles is typically less than 1 micrometers. So it's really kind of weird size ranges. It's not that small like molecules or free-floating proteins, or it's not that big like cancer cells. So it falls in some kind of gray zone in terms of the analytical method. So in other words, we need some method that are optimized for these size ranges, 100 nanometers or at least less than 1 micrometers size ranges. That is because when you think about the sensor, one of the key considerations in the sensor design is actually maximizing the filling factor. So by definition, roughly speaking, filling factor is the ratio between your sample volume, the target sample volume, and the detection volume. And we want to maximize this filling factor to achieve the best accuracy and precision on that sensing system. And here is the good example here. So let's say in the highway, on the ramp, there is this weighing station that can actually measure the weight of this big trailer or big truck. And for this, you need this big scale, as shown here. If you want to measure your weight, your own body weight, we don't use this kind of scale. We use this kind of bathroom scale over here, right? Let's assume that we switch the scale. We use this bathroom scale to measure the weight of the truck. Wouldn't work, right? The scale will break down. If I use this kind of scale for a truck to measure my weight, well, I'll be very happy because I might be 0 kilogram on that scale, or might be really sad because I might be about 0.1 tons, right? Think about this. We are measuring the same physical property. In this case, weight. But depending on your sample size, whether it's a truck or it's a human body, you need to choose the sensor that has almost the comparable the size scale. And that is what I meant by maximizing the filling factor. For nanoscale objects such as exosomes or other extracellular vesicles, the sensor itself should have the sensitivity in the nanoscale area or nanoscale volume. In that way, we can maximize the sensitivity. In other words, if I use the device that are optimized for cell detection, flow cytometry, for example, use that to measure the individual EVs or extracellular vesicles, might not get the good sensitivity. Okay, so that's the one consideration when you're designing any kind of a sensor, whether EVs or cells or proteins. So based on that principle, we have generated these different types of platforms optimized for the EV isolation and analysis. Again, here the key, the driving principle is make it smaller. The sensing volume smaller and smaller, so that they are comparable to the EV size. Okay, so these two are isolation device in the early design. So we have used the filter or even sound waves to isolate EVs, fluidic chips for PCR, and this is our single EV analysis or C technology, such that we can look at the individual EVs, protein profiling. On the bottom row, you are looking at the protein devices, the devices that are developed to measure the EV proteins. There is a microNMR early version, nPLEX, which uses surface plasma resonance, and iMEX IKEA, which uses the electrochemical sensing as a detection mechanism. So about 3 years ago, we published this review article in the Chem Review looking at the different types of EV sensing technologies. And if you're interested, you can look at this review. And then after that review, actually, there are now the kind of burst of the new platforms developed for EVs. So you might also want to look at the other technologies that are available in the field. So before going into the actual sensing method, I just want to emphasize or highlight why we are measuring EB proteins. Of course, we can measure the EB messenger RNA or even the DNA associated with EVs, but our primary goal has been to measure EB proteins. And the analogy for the importance of the protein is coming from this kind of online game, the experience. So on the top left, so he's one of the best online game player in the world. This boy is in Korea, but he's the world champion in playing the game called StarCraft. Some of you guys might know this game. I mean, you build this castle or the structure and then you fight each other, right? He must have a game plan before he starts the game, right? Or any of us who are playing the game, so we have a game plan in our head. But when you're actually starting playing the game in the field, the game plan is not necessarily equal to what you have or what you had in your mind. In other words, game plan and what's happening in the field, they're not exactly equal to each other. And that analogy can be translated into the tumor or biology as well. I can say that the game plan is kind of similar to the genotype. What is happening in the field is a phenotype, right? The genotype can tell us the static indicator of the risk, whether that person has a risk of developing diabetes or developing tumor or developing Alzheimer's, right? Phenotype is what's really happening there, the dynamic indicator of the status. And one of the good indicator or good marker that we can get the status is the protein. So that's why in our research, we have focused on detecting the EB, the protein, for cancer diagnosis and also the treatment monitoring. Okay. So with that introduction, so here are 3 types of device. Because I know that I don't have enough time to introduce all of these 3. So let me briefly summarize each of these devices. On the top, we have this nPLEX system. It's called nanoplasmonic exosome system. It has a really good sensitivity, exquisite sensitivity coming from the surface plasma resonance, and also has a high throughput. So we think that this device for this kind of system is well suited for high throughput or massive screening for the pharmaceutical companies or big hospitals, for example. And maybe because of that reason, so we teamed up with Exosome Diagnostics. So they have licensed the technology, and then they have actually produced this automated reader system for this nPLEX. So you can load this kind of chip over here, and the system will automatically read out the signal from each detection site, more than 100 detection sites. Okay, so that's the high throughput, high sensitive system. On the other hand of the extreme, we have a very low throughput research optimized system, which is C, or single EB analysis. It has a exquisite specificity because we stain the individual particles, individual EVs, and then we look at them under the microscope. Okay, so as you can imagine, this is very labor intensive, but we can get the protein information on the single EV level. So this will be a great tool actually for the discovery mode to find out the best markers, for example, for cancer detection. Okay. In the middle, now we have something in the middle. Not that high throughput, not that labor intensive, but still good enough for routine use in clinics or in the lab settings. And that's where this iMEX technology comes in. So iMEX stands for Integrated Magneto-Exochemical Technology. It uses the electrochemical sensing. It's fast, sensitive. I mean, it's less sensitive than the the plasmonic system, but it's still way better than the conventional ELISA. Cost effective, a small device. And so this is the system that I want to highlight in this presentation. And in my lab, as of now, I mean, this is the workhorse for EV protein analysis. Okay. Oh, by the way, so that system, we just published a paper using that technology. We look at the extracellular vesicles in the colorectal cancer patients. I will share this data with you in this presentation. So let's look at the technology first, the sensing technology. So this iMEX technology is based on the magnetic pooling. So we are using magnetic beads decorated with antibodies. So we just mix this magnetic bead with a sample, serum or plasma or urine samples, and then we just fish out our target. In this case, extracellular vesicles. We typically use the antibodies against CD63 or CD9 or CD81, those pan-EV markers, to capture these EVs on the magnetic beads. And then we just use magnets, not centrifugation, we just use a magnet to pull down these magnetic beads, do some washing, and then we further label these captured extracellular vesicles with probing antibodies. So if I want to measure, for example, epichem, then this will be the antibody against the epichem. If I want to measure EGFR, then this will be the antibody against the EGFR. So here you're looking at the SN picture of a magnetic bead after capturing those EVs. So you can see them on the surface. And then we are generating the analytical signal. So these probing antibodies, they also contain this enzyme, HRP, the horseradish peroxidase. And then when we mix this bead EV complex in the substrate, is called TMB, then there is this enzymatic reaction, electrochemistry basically, and then it will generate electrical current. The magnitude of the current will be proportional to the amount of antibodies on the bead surface. And of course, that amount of antibodies will be proportional to the expression level of that specific target on the EVs. So we can do the quantitative profiling of that protein level in a given sample. Okay, and the signal will be read out by this electrode in the form of the electrical current. So the assay is pretty simple and straightforward. Again, we just mix our sample with magnetic beads, capture the EVs, and then do the further labeling and put them on the electrode. We can measure the signal. So entire assay from start to the end, meaning starting from the plasma and getting the signal, is usually less than 1 hour. 15-minute EV capture and then 30 minutes EV profiling. And here on the right-hand side, we are comparing the sensitivity of our method, the iMEX, against the ELISA, the conventional ELISA. So iMEX titration curve is here, ELISA curve is here. So our sensitivity is close to about 10 to the 4 or 10 to the 5 EVs in the given per milliliter in terms of concentration. And usually we use about 10 or 20 microliter of plasma samples per marker when you're using this iMEX system. Okay, and also the assay time is less than 1 hour, ELISA about 3 or 4 hours. Okay. And in terms of the engineering, so I'll just flash out 3 more slides on the device. So whenever you are measuring the electrical current, not the optics, electrical current, it is very easy to miniaturize a device, make the device smaller, as well as perform the multiplex or parallel measurement. That is because we have so good IC technology, integrated circuit technology, that are way faster and way sensitive than most of the biological process. So in our first generation of this iMEX system, which is shown here on the top left corner. This is actually a photo of the device. So we have eight electrodes, 1, 2, 3, 4, 5, 6, 7, 8. And the size of the detector is about the size of a smartphone. So we just plug in this electrode, and this system will measure the signal one at a time from each electrode. So for each measurement, it will take about 1 minute, which means when I plug this electrode, it will take about 8 minutes to read out all the signal from the 8 electrode. Okay, so this is our pilot system, the first prototype. And then we saw that, well, there is a room for the further miniaturization. We can make this way smaller because when you open up this box, actually it's almost empty. I mean, there are some components, but most of them are just empty. So we integrate more, put more the functionality in even smaller the footprint. So this is our keychain device. It has a display, Bluetooth, mini USB, microcontroller, even the rechargeable battery. We can recharge this battery through the Bluetooth from the phone. So it actually it's a kind of parasite, and you suck out the energy from your phone through the Bluetooth. And this is a really small device, keychain device. And actually we developed this system to detect the allergen, peanut allergen and egg proteins and milk proteins on the restaurant or at home settings. But anyway, it's the same principle. We use the electrochemistry. Okay. The latest system that we have used in our colorectal cancer study is actually scaled up because we have to measure many markers to improve our diagnostic accuracy. So here we use the form factor that is compatible with our familiar 96 well plate format. Because with this kind of plate, we can use this multi-tip pipettes to easily load our samples or antibodies or beads or washing steps, right? So we designed this reader that is compatible with this 96 electrode array, and also put some interface over here, and such that we can measure the signal from this 96 well plate. One engineering innovation, the key innovation in terms of engineering in this device is that we measure the signal from all the 96 well plate at the same time. In the previous version, when I showed you that 8 electrode device, I told you that device measured 1 electrode at a time, and each measurement 1 minute. So you have to wait 8 minutes to finish up the measurement. In this system, not like that. It's not like we have to wait 96 minutes to get all the data. No, this system fast scan every single electrode, and they can finish the measurement in 1 minute because they kind of last scan. They look at every single electrode. It's so fast, and so it is as if this system read out the signal simultaneously from all the 96 well plate. So once you load the sample, in about 2 minutes, we get the result. So that's on the key innovation, engineering innovation in this system design. That is possible because we're using electrical measurement based on the electrochemistry. Okay, so that's the device part of this research. And then let me talk about the clinical applications that we have used with this device. So we applied our system, the iMEX system, to look at the EV protein markers in the colorectal cancer patients. And one of the big motivations was that for the colorectal cancer patients, the 5-year survival rate is actually very good. It's more than 70%, and more than 90% if it's a stage 1 when they find this tumor. And that actually, this long good survival rate gives us the opportunity to follow these patients over time. In other words, follow these patients while this patient is undergoing the surgery, the therapy, and also doing the follow-up. So with this colorectal cancer patients, patient samples, now we can ask the question, how does the EV profiles, the EV protein patterns, will change over time as a result of the surgery and also the drug therapy, the chemotherapy, and how they match up with this survival rate. So this using this colorectal cancer actually give us the window, this opportunity for this long longitudinal study. So that was one of the big motivation of using CRC in our EV study. Okay. So then the first question is, okay, I know the motivation. So we want to measure the colorectal cancer EVs, one markers. So first we have to decide the markers that can define the colorectal cancer derived EVs. And for that purposes, so previously we rely on the literature search or the experience from surgeons or our own, the lab know-hows to select the markers. But then that was not systematic. So we established this pipeline, bioinformatic framework to select the candidate markers for a given tumor type. And now we are using this publicly available databases to get these initial candidates. And here is an example for this colorectal cancer cases. So we use the human protein atlas, get the protein markers. This is so this database will give us the markers that are overexpressed in the tumor tissue and also expressed in normal tissue. So we compare those markers and then we select the markers that are overexpressed in the tumor tissue. In this case, colorectal cancer. And then we use this database, UniProt, to filter these candidates such that we choose the transmembrane proteins. In other words, proteins that are expressed on the cell surface. Because in our assay design, we are measuring the proteins on the EV surface. So that's why we filtered our initial candidate through this database to choose the markers that are on the cell membrane, the surface. And then we further use the EV database. So these markers will pass through this EV database, and then we further narrow down the markers that has the evidence that either reported or through the literature search that are present on the EV surface. Okay, so through this kind of framework, we can now narrow down from about 3,000 proteins to a handful of proteins that are potential candidates for EV profiling. In this case, the colorectal cancer. So these are after this kind of framework, so we have these markers, and some of them are well known for colorectal cancer. For example, EpiChem and GPA33. From these candidates, the next step is, of course, then we are using the cell lines, the colorectal cancer cell lines, and also EVs from human plasma, the normal plasma, to further validate that our candidate markers are really present on the cell line derived EVs. So in this heat map, what we are looking at is on the x-axis over here. So these are the target markers that we have identified. Y-axis, colorectal cancer cell lines, and this is normal control. And then we basically isolated the EVs and then look at the expression of these markers. And some of them are screaming hot, right? For example, CD24 is already here, EpiChem already here, EGFR is kind of good here, and GPA33 is also good. So after this profiling, confirming that our markers are present on the EV surface. Then we perform this for the statistical regression. It's called the loss regression here, to pick up the best marker sets. The best marker sets that can differentiate CRC, the colorectal cancer, versus normal. And in this exercise, this loss regression produced 5 marker sets: the EpiChem, CD24, EGFR, CD133, and GPA33 as the CRC EV markers. Okay, so far the in silico analysis, bioinformatics, and then cell line analysis. Okay, we need to verify that they are really present on the tumor samples and also tumor-derived EVs. So as a pilot study, we looked at about 11 patient samples. The samples were precious because we have blood samples and also matching tumor tissue samples. And here, these patients, on the tumor tissue, we have a high expression of the EpiChem, not much of EGFR, and the good expression of the CD24. Patient number 2, again, the very high expression of the EpiChem, high expression of EGFR, and also high expression of CD24. Okay. Now, when we analyzed EVs in blood, okay, when we analyzed the EVs on the matching blood samples for patient number 1, high expression of the EpiChem, almost nothing on the EGFR, nothing, almost, yeah, but negligible CD24. For these patients, almost all the markers, EpiChem, EGFR, CD24, they're high. So when you perform this analysis about 11 patients, we get the correlation about 80%. Not exactly 100%, but still there is a good correlation between the expression of these markers on the tumor tissue and the expression of the same markers on the circulating EVs, which really support the hypothesis that the EVs that we are looking at are likely coming from the tumor tissue. Okay, so we evaluate the markers, and also we chose the markers. Then the second question is, okay, you have markers, so what is the accuracy of this EV protein assay for cancer detection? We should be able to differentiate cancer versus non-cancer cases. So here is our first training set. So first cohort, 25 non-CRC controls. Some of them healthy and some of them have the irritable bowel syndrome, and some of them have the pancreatitis, not pancreatitis, other bowel disease, but they are not CRC patients. And also we have 58 CRC patients. Collect the blood, and in the plasma, we look at the expression of our 5 markers on the EVs using our iMEX system. On average, cancer patients, they have cancer cohort, they have a higher expression of the EpiChem, EGFR, all those markers, basically CD24, CD133, GPA33. That's the average when you're looking at the entire population. For each patient, the marker expression is of course heterogeneous. Some patients have the high expression of 4 markers here, for example. Some patients have the high expression of only one marker. So in other words, for the diagnosis, for the cancer diagnosis, we need the combination of these markers. And finally, we define the score based on the expression of the 4 markers, the EpiChem, EGFR, CD24, and GPA33. We realize that CD133 is not actually necessary. They wouldn't add more in terms of the diagnostic accuracy. So we have defined this EV CRC score, which is basically the average or weighted sum of expression of these 4 markers. With this EV score, now we can define the cutoff for the CRC diagnosis. and then this is a cutoff, and this is the RBC curve. Of course, when we use this, the integrated score, we have a higher the accuracy. But this is for the training set, right? So we have to validate that this same cutoff can be applicable in the validation sets. So in the validation cohort, another 33 CRC patients and then 15 non-CRC patients, and we measure the EV score, basically the weighted sum of those 4 markers, apply the same cutoff over here, and we can see that we can maintain still a good accuracy in the cancer detection. So overall, looking at, summarizing this, the diagnosis work, EV-CRC, 98% sensitivity, specificity is about close to 100%, and then 99% accuracy. In comparison, the conventional serum markers for CRC monitoring, which is CEA, has a sensitivity of the 76%. So we look at the same cohort, the same patients cohort that we have look at the EVs. 91% specificity, and the accuracy is about 80%. So the EV-based profiling clearly in this cohort, outperform the conventional markers. Okay. And then, okay, so that's the diagnosis. We asked the question, okay, what will happen on these EV profiles or EV levels after the surgery, after removing the tumor from patients? So here we look at the collected blood, about 13 patients before and after surgery, usually within 1 week after surgery. And first we measure the EV concentration on those patients, and also we perform that the EV protein measurement, those 4 markers, and then we calculated EV-CRC score. Let's look at the right-hand side first. All the patients that we have looked at, 13 patients, EV-CRC score, they went down. In this case, no exception. They just went down. And this is another second kind of strong suggestion that what we are measuring through this protein profiling or molecular profiling are the EVs that came from tumor. After surgery, tumor is gone, so that's why the score got smaller as well in this case. EV concentration, though, is a different story. In this cohort, you can see that statistically there's no difference between before and after surgery. And that is actually no surprise for us, because when you're measuring the EV numbers or EV concentration, we typically rely on one of these devices. The nanosite, they use the light scattering, or other method that basically count the number of particles in a given sample volume, right? So yeah, I got this clip from the Melbourne website. So we are measuring what is basically being floated, in this case, scattered by laser light. The equipment itself, measurement itself, there's nothing wrong in this measurement. It is just the interpretation of the result that got us the confounding factors. Because when we just measure the count of numbers in plasma, even after some purification, such as size exclusion, the chromatography, we will overestimate EV numbers. This image, this TM image, is coming from one of our samples. So here, the plasma samples, we purify the plasma samples through the size exclusion, the chromatography. And when you look at when you actually look at these samples on the TEM, I found one EV-like particles over here, right in the middle. The rest of the particles, they're LDL, low density lipoprotein, or VLDL, very low density lipoprotein. So the size of this protein is almost same as the EV, about 100 to 200 nanometer ranges. So when you just count the numbers through the particle counting method, you will also count these LDLs or other lipoproteins. And also, the number of these guys, LDL, they're way more abundant than EVs in plasma. About 1 million times more of these particles in the plasma. And that may be the reason that when you are just counting the EV concentration, we are not able to see the difference between before and after the surgery, because our EVs were basically outnumbered by these lipoprotein particles. But the molecular profiling is the key to look at or to measure the actual EVs from the tumor. Okay. So yeah, I digressed a little bit. Going back to the longitudinal monitoring here. So we look at the EV levels before and after surgery. And then as patients underwent the chemotherapy, and after the chemotherapy, we look at the EV score again, about 3 or 4 months later. These 4 patients, the tumor didn't come back, and then there is still the decrease of the EVCS score or the kind of stabilization of this EVCS score. In contrast, in those patients, the tumor came back, this EVCS score increased compared to the baseline, in this case, after surgery, right? So before surgery, after surgery, they all went down after surgery, and then tumor recurred, and this EVCS score started to increase over time. So this EV profiling now gave us the indication or inform the tumor recurrence. Not only for the tumor diagnosis, we might have this EV profiling have a chance to be used for the treatment monitoring. Of course, the sample number is low. I mean, it's about 11 patient samples. We need more data points and also more patients to get a statistical the significance. That is our ongoing work in this research. And finally, the final question is that we ask is, can you use EV levels to predict the patient survival? And ironically, this research was possible because of the COVID, pandemic. So we couldn't get the antibody, we couldn't get the samples. And so we've been just waiting. And then it happened that it already passed 5 years after we get the first sample from patients. So now we can measure the 5 years survival of those patients. In this case, we had 90 patients that we used for our training and validation cohort. So here we use the EV profiling levels, EVCS score, before surgery, preoperative EV score. And then we ask the question, how does it correlate with associate with the disease-free survival? And of course, what we have found is that when the score is high, in this case, the cutoff is 53, then the patient has a poor survival, versus when the score is low, the patient has the high chance of the disease-free survival. So this is still a pilot experiment, but what we are pushing here is going over the diagnosis, extending the potential clinical utility of this EV profiling. Okay. So summarizing this work, this specific work, so we developed this, the iMEX technology or iMEX technology that really enables the fast and high throughput EV protein analysis without ultracentrifugation, directly from the plasma samples. And in our assay, the molecular analysis or in the protein profiling was the key to detecting the tumor-derived EVs, not just the counting. Okay? And finally, our EV profiling in the CRC cohort, really outperformed the conventional blood markers such as the CEA in CRC detection. And it's still kind of pilot, but it apparently EV profiling informed the tumor recurrence and also has a potential, show the potential for the prognosis. Okay. And then wrapping up with a little bit bigger picture of what's going on here is that Analyzing these extracellular vesicles, extracellular or EVs, really promise a new era in the cancer management, diagnosis, monitoring, prognosis, and also maybe in predicting the treatment responses. Okay? But as I mentioned in the beginning, developing a right tool for the EVs, the nanoscale, these nanoscale particles, is very critical to get achieve the good accuracy and also realize the clinical translation of these assays. And also finally, there's a need to go beyond the diagnosis. Most of the articles out there or reports focus on the diagnosis. Now we need to extend the use of EVs for the other cancer management purposes, treatment monitoring, prognosis, and also, as I mentioned before, the predicting the treatment responses. That is actually to fully recognize and also exploit the advantage of the liquid biopsy, which is we can perform this longitudinal or serial profiling with a minimal complications. Okay, with that message, and I want to thank all the people in my group members who worked, especially Chongmin Park, Alajo, and Xingning Ning worked on this iMEX project. Dr. Weisletter, the director of my Center for Systems Biology, my clinical team, my exosome collaborators, and also the funding agencies. So with that, let me stop here, and I'm happy to answer your questions. Should I stop here?
All right. Thank you so much, Hakho. It's a wonderful presentation, and it's quite interesting and exciting new technology that we can use for the extracellular vesicle. So there's already a few questions here, and I think I'd like to start with Phil, because Phil has a couple of questions. Phil, can you unmute yourself and ask directly to Hakho, please?
Yeah, Dr. Lee, that I don't know. I'm on the phone and the screen.
There's a little bit background noise, but that's okay, Phil. We can still
No, well, because there's a storm here, and the internet connection is not very good. Can you hear me now?
Yeah, please go ahead.
So that's beautiful stuff, Dr. Lee, and obviously.
Oops. I think Phil suddenly got frozen.
Well, and what percent of the EVs that are CD63 positive are from the tumor? What percent of the number that are in the wells?
That is a really good question. So, roughly speaking, so that's we got this data from our single EV analysis. So about 30% of our EVs are CD63 positive.
What percent?
About 30% of EVs.
30%?
Yeah, about 30% are CD63 positive.
Really?
And so they're not really PEG EV markers, actually. They're
is that by flow cytometry? It's 63% or?
Oh, that is through the single EV imaging.
So I guess that's the other technology, the SEA.
Okay, so that 30% is what you're going to end up with in the wells.
Yeah, so so in our case, so we have actually use the mixture of the bees that has the CD63 antibodies, CD9, and CD81. And with that mixture, we can cover about, I couldn't say 100%, maybe
so
70
that's giving you 100% then.
Oh, not really.
Well, a high percent.
Yeah, higher than 30%.
Phil, I think you're frozen there.
Yeah, I got.
Yeah.
Can you hear me over here?
Yeah. So your second question?
What was what?
Yeah. How many EVs are there per nanowell?
So
No, it's all the same thing. How many per nanowell, and what percent are from the tumor?
yeah, so I don't know the exact numbers. I can give you the concentration. So it will be about 10 to the 7 to 8 EVs per mil. That's the concentration on the per well.
But what is it per well?
So yeah, that's the concentration per well. And then the sample volume is about 10 microliters. So 10 micro, 10 to the minus 2. It's about a million EVs per well.
Of from the tumor?
Not necessarily all from tumor. The tumor-derived EVs will be much lower than this.
In the patients, not from a line, but
yeah,
from
in the patients,
blood
yes.
of a.
That was, yeah.
Yeah, so I think that it's very interesting approach that you're looking into the disease first, rather than I think the majority approach is looking into isolated vesicles and rely on a lot on the EV markers.
You mean, let me rephrase the question. So whether EV markers are good for
because, yeah, so because currently, if we want to look into extracellular vesicle samples, one way of confirming that what we are looking is truly extracellular vesicles is based on the markers that are mainly the tetraspanin, CD9, CD63, CD81, and a few other things. So now with this approach, it seems like it's possible, or would it be possible that we also looking at non-EVs in the assay that you're using?
Good point. Now I got it. Yes. So in our assay, our assays is more like fishing, right? We fish out EVs based on the CD63. And that's why we are a little bit more confident that the epichem that we are detecting are on the EVs. Now, the question is, okay, if I just use human plasma and without any purification, with some purification, such as site exclusion chromatography, with that purification, and if I detect certain markers, are they coming from EVs or are they coming from, let's say, the lipoprotein? That we need to be more careful. I don't know the answer. I'm not. I really don't know whether LDL contains epichem on their surface. Highly unlikely, but unless you really isolate LDL and then measure that for sure, we don't know. So what I'm saying is, we say that we measure the EV proteins in plasma, but let's make sure that we really have on EV population when they're doing that measurement in plasma. Yeah.
There's also a lot of report that suggesting that EVs cargo, they are not mainly expressed in the tissue. So I'm just wondering whether you have a comment also on that, because the approach also looking into the tissue database. Whether that's just it's hard to say. What do you think about it, Hakho?
Yeah, so that's a good point. So initially we thought that EVs are really it's like an avatar of the tissue, the cells, so they might have the same expression of the markers as in the cells. some of the markers, for example, EpiChem and CD24, we found that that's true. It's really good correlation across the cell lines that we have used: colorectal cancer, pancreatic cancer, ovarian cancer. Other markers, not really the case. One of the key markers that hits me was the CD44. They're not that highly overexpressed in cells, tumor cells, but they are screaming hot in the EVs. So you're right. We cannot say that this is 100% correlation between EVs and the cells. Yeah, there are certain markers in which.
In terms of biomarker, your assay definitely shows a very promising techniques to look into whether patients are recurrent or define whether they are cancer patients or normal patient. I think it's really great. So the next question, Camille, can you turn on your video?
Hi, so my question is regarding with the non-CRC samples. Are those, do you have other types of cancers in that subset? Because I was just curious, because CD24, EpCAM, and EGFR, they've also been shown in ovarian cancer EVs to be present.
Yes, that's a wonderful question. Short answer, no. The reason, because there will be a confounding factor, of course. I mean, so this assay, the CRC assay, is designed to differentiate colorectal cancer versus non-cancer, not differentiate tumor types. We are doing some ongoing research here. That's tough. That's tough. Yeah.
Yeah. So yeah, and like I just want to give some input, like with the CD63 and CD81, like it's also possible. Like, can you see it in the SEA if like some of them are double positive for, like, say CD81 and CD9, which usually happens?
Yes. So of course, so that's the single EV analysis, and at that time we look at the GBM cell lines actually. So EVs from brain tumor cell lines, and then we analyze. They're all isogenic, meaning different cell types, but coming from the single cell, and they develop this certain mutation, and so it's called isogenic. And when you look at those EVs, we look at the 14 markers on those EVs: CD63, CD81, CD9, EGFR, EGFR-V3, IDH1 mutation, and so forth. The majority of the EVs, they have 2 or 3 markers, 4 at most. Most of the EVs, they have only 2 or 3 markers of the 14 markers that we have looked at. So there are certain EVs that have CD63, CD81 co-expression, but it's not like 60%. It's more like, I'm just making up this number, 50%, less than 50%. Yeah, but my point is, not many different protein on the EV surface actually.
Okay, thank you.
Okay, so I've got a couple of questions also on the chat. One is from Diana Shi. She's saying that, thank you for the very interesting talk. I'm wondering if there's a limit of detection for this nano assay, referring to the volume of patient samples. I think it was 10 microliter, was it, Hakho?
Yeah, 10 microliter of the plasma, yes.
Yeah. And last question is from Ayesha Salim. She really, and I also really want to know whether you are collaborating to share the iMEX technology, or is it commercially available?
Oh, so there's a local startup, so they license the technology, so I think they'll do something about it. But I want to say, at least for the iMEX, meaning using the electrochemistry for EV detection, it's very easy and straightforward. Okay, if you don't mind the several pipetting, it's better than, yeah, I cannot say better, but it's better than ELISA. Yeah.
Okay, more questions. Masha Jalali, can you go? Yeah, please turn on you're muted.
Hi, thank you, Dr. Hakho Lee, for very fantastic talk. I was just wondering whether it's possible to actually determine the percentage of the tumor EVs in a pool of the non-tumor EVs and LDLs with the iMEX technology.
Good point. So, for example, LDL, the percentage of LDL, we can use the antibodies against ApoB, and using the iMEX or iMEX technology, then we can estimate the amount of the LDL in given samples. Actually, but I wouldn't recommend that if we're using SCC, because even after SCC, we have a huge number of LDL. So it will just saturate your detector. So you can just use any bulk method to measure the LDL. Depending on the tumor stage, right? The severe patient, they have more tumor-derived EVs. Just a rule of thumb, just rule of thumb in a sense that even for the late-stage patients, it's about maybe 5 or 10% of the total EVs are actually tumor-derived. It looks like, but it's not like 90% of the EVs are coming from tumor, because that doesn't make sense. In our bodies, they all generate EVs, right? And it's in circulation. So, yeah, tumor-derived EV is about, I would say, safely speaking, less than 10%. Yeah, so now you see that this is challenging, right? Got LDL, EVs, and then tumor-derived EVs. Yeah.
Yeah, I think, would it be possible to actually assess the LDL itself? I mean, there are some markers for LDL, so maybe if you can add that into the assay, and then we can sort of exclude those.
Yeah, so our strategy is actually to, so we are working on this project right now, is we even want to separate the LDL out from our sample after the SCC, because the LDL and EVs, they have different surface charge. So now we can use charge, not only the size, not only the density, charge as another handle, another axis to tease out EVs from LDL. And with that method, we removed about 99% of the LDL from the plasma.
Oh, that's really cool.
But still, 99%, but it's still way more than, I mean, LDL starts with, there are way more than EVs, so still we have a little bit of contamination, but it's better than SCC, yeah.
Yeah. Okay, I think that's pretty much all the questions. So with that, I'd like to thank Hakho Lee for your presentation today, and very interesting and promising tools for us to actually analyze the extracellular vesicles in blood. Actually, I want to know, do you process the blood at all apart from getting the plasma? Do you actually do some sort of like steps before using your assay, or you can directly from plasma, like dirty plasma?
Just plasma, just plasma.
Yeah, that's very cool.